Hello. In this video, we're going to discuss how to identify the main genes responsible for your cluster's differentiation. So for that, we have a dedicated block, which is called cluster markers, and I assume that you already have your data processed up to the point when you have clusters ready, for example, using Leiden Clustering block. Here, I'm going to add the block, which is called cluster markers, to the project. And now let's discuss the settings. So the first one is obvious. It's your data set. So here you see cluster resolution 0.5. This is the output of Leiden clustering block. Top markers per cluster. This is the number of genes that you will see on the dot plot here when we run this block. So this number does not affect the way clusters are calculated or differential expression is calculated. It's just the way to how to visualize your data. How many genes do you want to see? And we're just going to leave 3 by default, but you can change it later. Now, this part is rather more important. Marker specificity mode. What does it mean? So non-exclusive means that only genes that are expressed by more than 20% of the cells in the cluster will be taken into account, but regardless of the expression of these genes in other clusters. Now, if you select strict overlap, what it does is that not only you're gonna pick the genes that are expressed by more than 20% of the cells in your cluster, but there is another condition that at the same time, the same genes should not be expressed by more than 20% in any other cluster. So this is a more strict condition, and if you want to narrow down your options, sometimes it's better to select strict overlap. And now finally, we are selecting the log two-fold change threshold and a p-value. So these values drive the identification of the differentially expressed genes between a certain cluster and all other clusters. So I'm going to click run here, and we're going to look at the output in a second. Just going to close the settings and wait for the block to work. So now we have the table. This is the table of all differentially expressed genes that pass the threshold, which we set for log two-fold change. Yeah, the threshold that we set here was 1, and p-value. So these are the genes symbols for cluster 0. You can select a different cluster and look at the different table. So just a table that you can export if you want and see the expression of every gene or, well, differentially expressed gene for each cluster. And also here, there's another tab which is called dot plot, which is a very common way how to look at the clusters. It's a dot plot of the genes that are majorly differentially expressed in each cluster. And remember, we selected 3, so we only put 3 main differentially expressed genes per cluster. So for example, for cluster 0, this is gene 1, 2, and 3, and you don't see anything else. Same goes, for example, for this one, gene 1, 2, 3, and then there is like nothing else for cluster 11. So for each cluster, you get 3 major differentially expressed genes. That is all for the cluster markers block. And in the next videos, we'll look deep into what you can do next with the single cell RNA-seq data. Thank you.